The question is, how does the band 3 protein causes glycolysis in the RBC in case of hypoxia? So understand one thing, that band 3 is one of the most abundant proteins that are present on the RBC membrane. Along with other functions that they carry out, one of the important function is that they allow site for the glycolytic enzymes to bind. Okay, so if this is the band 3 protein, these are the glycolytic enzymes that are bound to band 3. When these glycolytic enzymes are bound to band 3 protein, they'll be in the inactive form. Now coming to hemoglobin, when the hemoglobin are in oxygenated state, that is when they are carrying enough amount of oxygen with them, they will not be binding to band 3. But in case when they are deoxygenated, like in case of hypoxia, so in this case, this deoxygenated hemoglobin will go to band 3, they will displace the glycolytic enzyme and bind themselves to band 3. And once these glycolytic enzymes are freed from the band 3 bond, now they'll be converted into active glycolytic enzymes. So we can say in case of hypoxia, the band 3 releases the glycolytic enzymes to cause glycolysis within the RBC.